online over there and stuff. It's all about what we go on every night. I was scared about that. Because there's things from the beginning of genetics that are, you know what I mean? So, I had to have a professor do that before.
Okay, wait 1 second. What? The recordings or the slides? Recordings on the YouTube. There's a link in the platform. When you click on the link, it should take you to YouTube. If the link is giving you trouble, let me know. Okay. As you guys all just witnessed, I'm not good at multitasking. But so first of all, I just checked the forecast because this is like this is very recent. Who likes Foot's forecast? Anyone? Yay! Foot's forecast.
So we're feeling it.
So I'm thinking Thursday might not happen. Foot's forecast is actually really good. But Thursday might disappear into the ether.
Okay, great.
And we'll just go straight for Wednesday. So I have a plan. I know you guys are so excited about a snow day. I hate snow days, just so you know. They're fun like once a year, but they really mess with things. So I have a plan. Okay. First of all, before I go into my plan, I wanted to clarify something regarding weekend quizzes. People keep saying, but you get 2 attempts. Why are you just recording the first one? The reason that it says there are 2 attempts is because I have it set to automatically allow you to take a second attempt in the event that there's some technical mishap. If there's a mishap, you email me, say, oh my God, my computer was, I don't know, killed by a cat. And then you email me and I say, go ahead and take the second attempt. And then I note that you took the second attempt, save that score, get rid of your first score. But normally, your first attempt is recorded. Why? Well, what's the point of me letting you take something twice? So you can look at all the answers and put them all. I mean, we all get 10s all the time, which I do want you to get. But, so that's the clarification. It says in the instructions, the first will be recorded because those are the instructions I write. It says there are 2 attempts because that's what Blackboard automatically puts because I have it set that way. Do not take the second attempt and expect to have it count unless you email me explaining your drama of some sort, and I look at your test and I see evidence of the drama and we're all good. That happens. It happens all the time, and that's why that's set up that way. Don't take it quickly and just submit it because you have all weekend, which brings me to the second thing, which is that I will keep quizzes open till Monday night from now on because someone brought up a point that, not really, actually, it was a point that came from a conversation that I understand you guys work all weekend. I try to get things up earlier on Friday, but I never can get as early as I'd like to. Since we don't have class till Tuesday, having them open on Monday is no big deal. So they'll be open through Monday night from now on. So those of you who are working from the minute classes end on Friday until they start again on Monday, you'll at least have Monday to get this done. Exam 1 is currently scheduled for the 25th. I did decide to move it to the 20th. Because I have 48 slides left and I do not need that much time to go through them. That's about 1 lecture's worth. I'm actually going to give a short lecture today. We'll have, Thursday's disappearing, so there'll be no lecture on Thursday. And then, we'll finish things up on Tuesday and then have our exam. So just plan for it. I think this is plenty of advance notice. So feel free to call me disorganized. That's fine. What else? If you're interested in an oral exam, please contact me, like now, at least a week in advance. Okay, So recitations, that room that I have my recitations in is not working for me or us or anyone. So I have finally figured out what to do. And I will send this out to you guys via email as well, because I know not everyone comes to lecture. But Tuesdays, 12:30 recitation is changing to room 275. Thursdays, all of my recitations, so that's 10 a.m. and 12:30 p.m., moving to 217. Those rooms are a bit better for recitation. Other recitations, the one other recitation taught by Dr. Texel has not changed. Do not go to a different room. Okay, so snow. Back to what's forecast, right? Snow. If there's snow and you miss recitation, this is how we will make it up. Basically, we will make up that paper next week for Thursday's group. Tuesday, who already had that recitation, that'll be a review for you guys. So the paper for next week will not exist anymore. So it's now the paper from this week. So don't go to next, and I'll put this all on Blackboard to clarify it. So, but we're making up this Thursday's paper next Thursday, and we'll still use the rest of the time for review. You guys will still have plenty of time to ask me Q&A about any remaining things. Now, yes, it is before the exam, so bummer. But that's what we're going to do. Now, note, we have a review session scheduled for the following week because that was the week the exam was supposed to be. That review session is being moved to next week. So there's no point in a review session after the exam, right? And so I'm going to have to mess around with the syllabus. So don't go ahead and print out all the papers in advance because these things happen, especially when you're transitioning your class from one schedule to another. So bear with me in the snow. So in the snow, I'm not the only disorganized one. The snow is also very disorganized.
It's a good disorganization.
Okay. One last note. There's a lot of business today. Recitation 004, which is Dr. Texel's recitation. Please plan to attend either the 10 a.m. or 12:30 p.m. recitation next Thursday because the review sessions will not be led by her because she's not teaching the class. They'll be led by me. Let me know if you cannot make either of those recitations. Email it. Don't let me know now. Email me and say, oh my God, I can't do that. I'm assuming since you can make it to her recitation, you should at least be able to make it to 12:30. So just keep me posted about that, and we'll make a paper together and do the review together. Big, happy family. Okay, is everyone clear with all those little details? Yes?
So that means next week, Thursday, it's not going to be at the regular time? Is that what the last one
Are
is?
you in Dr. Texel's section? Then you will come to one of these instead because unless you want her to leave your review session, but it's not really ideal since she's not teaching the class. So I told her, take a break. I'll take the everyone so that you guys can all benefit from me being in the room. Okay? Let me know if it doesn't work. Okay. Everyone good? Yeah.
So next Thursday is our first exam.
Next Thursday is your first exam. Are you excited? Just get it over with. Okay, so let's learn something. We just got to get it all over with. We got a minute.
Sorry, so there's 4 labs in this class?
There are 4
Okay.
recitation sections.
So I'm in the 1 o'clock one, so you do 12:30 instead.
Yeah.
Yeah, next
If you
Thursday.
see Dr. Texel and not me during recitation, come to one of these 2 instead. Okay?
And will we do this, if we have a snow day, will we do this paper for this week and review?
Yeah.
Okay.
Exactly. If we have a snow day, then we'll do both. If we don't have a snow day, which I really think is highly unlikely, but if we don't have a snow day, then you'll do that paper this week with Dr. Texel, and then you'll still come to my review next week because I would like you to have me for the review. Okay? Okay. Happy? Everyone's feeling good?
Yes, we have a review right before.
Okay. Yes, you have a review right before the test, so all your last minute questions can be answered. Okay, this is your study guide so far, right? Basic big picture idea of the cell is a part of a larger organism that needs to respond to the environment. That's what it's all about. The different methods, do not know them in terms of first you do 5 microliters of this, then 5 no. Just when would you use them, when would you use them? Chromatin structure, nucleosome structure, post-translational modifications found on histones and the protein domains that recognize them. Those are your acetylation, methylation, that kind of thing. The general function of post-translational modifications. General concept of protein domains. Histone acetylation and the enzymes and the associated function, so everything about that. Histone methylation, associated enzymes and functions. Histone code, the concept of a histone code. Chromatin remodeling, and histone variants. That is what we have done so far. I will keep adding to this list. But this so far is the topics that you need to know. Okay? Okay, so this is where we ended the other day. And someone pointed out that it was not a perfectly controlled experiment. So I decided to show you this. See, this is why I can't just take my magical slides out of a desk drawer because I have to respond to what happened the time before. So the way you would see so this is, let me orient you. This is just taken from a paper. This is H2AX, so antibody against H2AX. Secondary antibody is green, which is why you see green. DAPI stains DNA. So your untreated cells, you see nothing, right? This is some sort of something or other they're treating their cells with. UV light, right? So again, it's causing DNA damage, Here they're combining the two, causing DNA damage. Ionizing radiation, again, known inducer of DNA damage. And so here you could say that gamma H2AX is only added to the chromatin in instances of DNA damage, right? So if you went back to this experiment that we did, then you would look again at gamma H2AX and be like, oh, it's all over the place. There must be DNA damage. It's a good cancer drug, right? So that's the corrected version of the practice problem we did. Okay. So that was the end of talking about the histone variants. Okay, so let's talk a little bit again about the histone code. So remember, the concept of the histone code is about just the different modifications that occur on the histones and how they are going to recruit specific proteins to do specific things. For example, there could be a certain code that will induce chromatin condensation. Right, so that's kind of condensing of the chromatin, bringing all those nucleosomes tightly together so there's no accessibility to the DNA. This is associated with what? Gene expression or gene or repression? Silencing, right. So, so chromatin condensation it's the spreading of this certain code throughout the chromatin so that its structure spreads. So this is through the collaboration of readers and writers. I think it's kind of cute. So in this case, you can see you would have some sort of gene regulatory protein recruited to the DNA under whatever conditions that are requiring chromatin condensation. And then it might recruit some enzyme that's going to modify the histone. If you're doing condensation, what kind of modification would you expect to see? What? Methylation, thank you. So then you get methylation, and then that methylation is recognized by one of these reader kind of complexes of proteins that's going to recruit some writer proteins. What kind of protein do you think would be the writer that's recruited if you're establishing repressive chromatin? Kind of writers being recruited. An HDAC might be one. What else? Huh? What kind of methyltransferase? I guess you can go with either, but Histone? Histone methyltransferase? Is that what you're saying?
I would say like something that has
Oh, right, yes, so deacetylase, so I heard HDAC over there, same thing, yeah. A histone methyltransferase or maybe a DNA methyltransferase, yep, exactly. So it's gonna recruit a protein that's gonna modify the next histone, which is then gonna maybe also recruit a protein, chromatin remodeling complex, which will help to kind of move your nucleosomes, pack them tighter, right? And then you'll get more readers and writers in chromatin remodeling. And notice you have this kind of more open chromatin, and here you're starting to kind of pack it up more densely. Okay? And so that's how you have this collaboration between your readers that are reading your histone marks and your writers that are spreading them to different nucleosomes to basically spread the chromatin condensation to create this repressive chromatin. Okay? Yeah.
So wait, what's the modification? Is being read?
I'm going to make you answer. What do you think? So if we're creating repressive chromatin, what modification do you think might be being read to recruit other proteins? So your choices are acetylation, methylation, phosphorylation. Right, right, because methylation is most associated with gene silencing, so it would be logical that that mark would be there and be recruiting more silencing type. Yeah.
Does it matter if it was a histone methyltransferase or DNA methyltransferase?
Right. Does it matter whether you're recruiting histone methyltransferase or DNA methyltransferase? As you'll see shortly, they kind of both do get recruited. Yeah, because they're both associated with that silencing. Yeah.
Reader and writer only work with methylation, or
Does the reader and writer only work with methylation or also acetylation? What do you think? So it really depends on what's being read and what's being recruited. Really, readers and writers are just kind of categories of proteins, right? So what if the reader is reading acetylation and recruiting demethylases? Why not? Or recruiting more hats?
so it's
Why not? Yeah, why not? So it can, why not go either way? Venus, what were you thinking?
Yeah, I was kind of had a similar question. I was going to ask if they could also read deacetylation to recruit. So you're saying
Right.
that it can.
So if there's a mark there, so that's the whole concept of the histone code. There's all these marks on your histones that are creating some kind of binding site for a complex of proteins that's going to recruit other proteins that are going to carry out the function that needs to happen. It doesn't always need to be creating repressive chromatin. It could be, more activating forms of chromatin. Yeah. Okay. Did everyone follow that little conversation? Okay. Any other questions? Yeah.
So when it's a reader and writer, it doesn't always have to be repressive.
Right, so readers and writers it doesn't have to be repressive. You have to think about it in terms of histone marks. There are different types. There's acetylation, there's methylation, there's phosphorylation, there's ubiquitination, there's simulation. There's all these different things, and they're all going to kind of create a very specific binding area for recruitment of some kinds of proteins that are going to do something. And that's why it's a code. You got lots and lots of methylation, you might end up recruiting this kind of complex that's used in this example. That's going to kind of spread this chromatin condensation. But maybe if you've got lots and lots of acetylation, it might be doing the opposite and recruiting the types of proteins that are involved in further acetylating and removing methylation. Okay, The other thing to think about is, you've got ubiquitination associated with DNA damage and that kind of stuff. So all of that's happening there is it's recruiting other things that are associated in recruiting proteins involved in DNA repair. So just think of it that way. Yes?
So the writer proteins are the ones that are doing things?
They're writing. So they're either acetylating or methylating. That's the writing. Yeah.
So those are the proteins that are being recruited.
Mm-hmm. Yes.
The nucleosome remodeling complex, its function is that it moves the nucleosomes around the DNA, and it kind of controls what's being expressed and what isn't.
Right, so nucleosome remodeling complexes are recruited by all of this, and that's moving your nucleosomes around, which is regulating the regions of DNA that are available for binding to other things. And so gene expression looks like you're getting it. I love it when I see like this little smile, like, oh, good. I want to see more of that. How about over here? Okay. Other hands? I saw other hands going up. Are we good? Are we happy? Okay. Sure. Okay, so regulation. So everything's about regulation with me. And so the question is, how do nucleosome remodeling complexes know where to go? So based on what we've just been talking about, it should be clear to you, right? So there are these transcriptional regulators that essentially recruit the nucleosome remodeling complexes through either direct DNA binding or through interaction with modified histone tails. And these are these chromodomain, bromodomain containing proteins that get recruited to these modified histones. Okay? So you've got you can think of them as more of like, you've got your readers, right, that are being recruited. And so most of it is really about recruiting the nucleosome remodeling complexes to move things around to expose different regions of DNA. And so you have to think to yourself, I really want you to start bringing it all together with what types of modifications are you seeing and what kinds of transcriptional regulators you would expect to be recruited to these types of modifications and what kinds of domains they might contain. Okay, here's a practice, or we're going to start out with a practice problem, I guess, if you can call this starting. So you're studying a novel protein, so novel meaning new, no one knows its function, that's expressed by some virus. We'll call it the XYZ virus. And you're trying to characterize the protein's function, and you have following information. You have the sequence of the protein, and you've blasted it. Everyone knows what blasting is? That's where you take a sequence and you compare it to all the sequences in GenBank, and you see how similar it is to other sequences. And you find that it contains two conserved domains, a bromodomain and an ATPase domain. Based on this information, what is a likely function of your protein, and what experiments might you conduct to provide evidence for your hypothesis? So take a minute to think, to discuss with your friends. And then when you're designing an experiment, I always want you to ask yourself, what is the goal of it? Because people always like to tag things and look at things. And that's like not the most direct way to answer everything. So I want you to stop and think to yourself, well, what have we learned already, and how do we know this, and that kind of thing. So take a minute to discuss. Yes.
There was like third.
Right. But if you don't consider the right, all you will know is
If the older and the older, you have the exact third. So like type if you want. I looked up stable
All your Okay, guys, lots of interesting comments. Are we ready to discuss, or do we need more time? Ready? Okay. So I got one question, which was, can we do anything related to the paper we read last week in recitation to determine this? Well, let's talk in a minute about that. Got another question about comparing to other bromodomain containing proteins, maybe from other viruses. Okay, so the first question is, you know that has these two domains. Based on what we've learned, what kind of protein might it be? Okay, I heard nucleosome remodeling complex and I heard hat. What makes you think it's a hat?
Because the separate domain is adds the distance to the end of the it adds itself to end of the distance, and it's using the ATP energy to build something.
Okay, that's interesting. The only thing that threw you off is that bromodomain binds to acetyl groups. So, and yes, it does use energy to do some process, to build something, to move something. Yes. Nucleosome remodeling complex. Why do you say that?
I said that because the bromodomains bind to acetylated histone tails, and nucleosome remodeling complexes are ATP dependent, so they need ATP to move the nucleosomes around.
Right. So exactly. Bromodomains associated with acetylation, so it binds to acetylated histone tails, and ATPase domain, that's going to be nucleosome remodeling complexes are ATP dependent. So that was a logical conclusion. So it's a hypothesis. So you're sitting in the lab, you see this, you form a hypothesis. So now we have two competing hypotheses in a way, but we've already established that one might not be ideal. But if we wanted to, we could develop two types of experiments to test them, right? So let's go with this first one here about, well, let's go with the nucleosome remodeling complex one. What type of, well, so you know it's going to remove it's going to move nucleosomes. And you know it's associated with acetylation, right? So is it going to be moving nucleosomes in order to create repressive chromatin or in order to activate gene expression? Okay, So you have some information here that can be used in developing an assay, right? So that's one thing. So I got a comment before about, well, can we do something related to the paper last week, Knoll's paper, where they were studying how much DNA is out there on each histone and degrading the ones between in some way to assess movement of nucleosomes. And so my question is, I don't know, maybe, because maybe, well, you know what, I'm thinking about your question. And had it had a chromodomain, maybe, right? Because if you have a chromodomain and it's associated with repressive chromatin, then you would imagine you would have less naked DNA, and then you could start to look at the lengths of DNA, because you should have longer lengths of DNA if you have more repressive chromatin, right? Then you had a question about, you had a thought about comparing with other genes in other species that have similar domains.
I was thinking maybe, like, not to last week's paper, but this week's paper, maybe more related to that.
So some kind of activity assay?
Yeah.
Okay.
To determine if it actually adds the acidyl groups.
But remember, what adds acetyl groups? Nucleosome remodeling complexes or HATs? So that would be the way to assess whether or not, what's your name again? Fatima is correct. Is you could put your protein in a acetyltransferase assay, like in the paper we read this week. Sorry for the 3/4 of you who probably haven't read it yet. And assess whether or not it is a HAT. And you probably would find that it's not, but maybe you would find that it is. And then, so then if you find that it's not, then you need to go to hypothesis number 2, that it's a nucleosome remodeling complex, and that brings us to how do we determine that? Yes?
Well, one experiment you can do is you can add a HDAC to, like if you had it in a solution, you can add the HDAC to it and see, once the HDAC removes the acetyl group, then the remodeling complex shouldn't be able to bind to the telling you which means the movement shouldn't occur.
Okay, so add HDAC, see if that prevents nucleosome movement. Okay. Yep.
Can you just look for protein expression?
Look for protein expression. Which kind of protein?
That's the specific genes he does.
Okay, so a gene, some kind of gene, just some kind of, not a true reporter, but some kind of type of reporter that could tell you whether or not gene expression is happening or not? Okay, maybe. Let's stay with that for a minute. Yeah?
Could you take DNA and supply it with the protein that you isolated and provided with ATP, and then after that exposure, if it is a nucleosome remodeling complex, the gene expression will change, and if you run a gel on that, you can see levels of expression based on your control.
So that's kind of similar to what you were trying to say? See whether, because you're talking about protein, were you talking about gene expression? Mm-hmm. Okay, so add your protein and see whether or not gene expression changes. So you would have to have some kind of reportery kind of gene that you would be looking for. Yes?
Can you like
Remove the protein, so like delete this protein and see. Okay. So that's another, so that bring this all together, and you can start to develop an experiment where you're either deleting, you can either have the whole virus and delete that gene from the virus and see, because probably it has this thing because it's inducing some kind of gene expression from the cell, right? So you can either delete the whole gene from the virus and see whether there's effect on gene expression, or you can start to delete these domains. You can delete your bromodomain and see whether or not it can bind to acetylated histone. Yeah, so this is how you do science, right? You generate a hypothesis, and then you start to toss around ideas on how to test your hypothesis, and it requires a lot of fine-tuning to figure out which experiment is going to actually answer your question. So we're close, yeah.
I have a question about the actual domains.
Mhm.
Are they both serving 2 different functions? One is silencing and the other
So there's two domains. One's a bromo domain, which is associated with what?
That's going to be gene expression.
It's associated with promoting gene expression because it's going to bind to acetylated histone. ATPase domain is associated, not silencing, that's chromodomains usually. ATPase is really, it binds ATP, hydrolyzes ATP into ADP to have some sort of process or movement occur.
Okay, I think I was thinking something wrong. I think I was thinking since I had ACE, it was like.
Yeah, no, yeah, people always, so ACE, you always got to pay attention to what's before the ACE to wonder what's being broken down. There it's ATP to ADP.
Okay.
Did I hear another hand? Yeah.
For the bromo domain, you said the addition of the bromo domain to see if it still binds to.
Yeah, because the job of a bromo domain is to bind acetylated histones, right?
Yeah.
So why not delete it from your protein and see if it will bind to acetylated histones?
But if you delete it, it can't bind, like, would the bromo domain do other things to bind?
No, so that's the thing, is you're proving whether or not it's acting as it should. Right, so normally when you're trying to ascribe a function, one of the main things you can do is delete things and see if they still act as they should. So if you think it should be a nucleosome remodeling complex associated with gene expression, you can start to delete things that make it a nucleosome remodeling complex. One might be delete the bromo domain, because that's very specific. And then you can look to see, is it binding to acetylated histone? Is it still able to induce gene expression or allow for gene expression? Okay, any other questions? Okay. Did people follow that, kind of, sort of? Yeah.
Just for clarification, we're going with nucleosome remodeling complex, which makes histones for gene expression.
We're going with nucleosome remodeling complex, which moves around histones, promoting gene expression. Yeah. And our experiment would somehow involve deleting either the whole protein or domains within the protein and assessing either probably a combination of interaction with acetylated histones or gene expression. Yes.
Just how an exam question go.
In a perfect world where I had 10 people in my class, yes. It would be, but I do like to have these kind of things where, but what it would be turned into multiple choice. Yes, this is similar type of exam question that I have because I'm testing, what I'm really testing here is, do you understand domains? Do you understand what a bromo domain is? Do you understand, can you pull all that information together and talk to me about nucleosome remodeling complexes? And usually I would provide a little background to kind of hopefully put you in the right direction. Okay. The other thing about my exams, since we have one coming up, is I encourage you to ask me if you don't understand a question, to come and truly ask me. And I've had people sit in the front row and ask me about every other question because they just don't understand my wording. And so, please, do that. Okay. Good. Okay, so try it yourself. Some of you guys jumped the gun and did this, so I've actually changed it. Some people last week were like, ooh, I'm going to do it. We're very excited and got it right. People emailed me their stuff, and I was like, we didn't do this yet, but that's right. But I put in a new one, so you guys can have fun again. So try yourself. What's the likely function of the protein below based on the domain organization? Basically, what you do is you take, this is protein sequence in single letter code. What you do is you blast it, like we did here. You blast it, and it will give you some information about the domain organization. You'll look at it, and I mean, you can probably cheat your way through it if you wanted to, but let me know what it is, and let me know what domains are there. Don't just tell me the protein's name. I want a list of domains and what they do and how you arrive at your conclusion. And so get it to me before Wednesday, 5 p.m., and you get an extra point. Bunch of blast things, isn't it? Now everyone in this class will be able to say they've blasted things before. Okay. If you don't know how to blast, go to NCBI. Feel free to ask any friends to show you how to blast. I'm sure Who here knows how to blast things? Raise your hand. Okay, there's a good number. Look around. Keep your hands up. All of those of you who don't know how to blast, look around at all your friends who know how to blast and get their phone numbers. Okay. Good. Okay, someone wants to go home. Okay, so let's talk about, so we talked about, we have trouble, there's always trouble in the front row. Always trouble in the front row. Trouble rarely sits in the back row. Okay, so what is the biological role of DNA methylation? So we talked about histones a lot. But what's the biological role of DNA methylation? Yeah. Reduce gene expression, silence it. Yeah, definitely in eukaryotic cells, definitely. Okay, very good. Does anyone want to add to that? Yes. Mismatch repair. In bacteria, oh no, now I'm going to question it in bacteria for sure, DNA repair. Is it in eukaryotic cells too, DNA repair? We just took cancer out. Can you answer that?
I think it was just to label like whatever your template strand was.
Right, your template. Okay, so repair during replication.
Yeah.
So you know what the correct strand is
Yeah,
versus
just kind
the
of
damaged strand. Huh?
Your undamaged strand.
Yeah. Okay. Your undamaged strand is methylated. So it's more that your old DNA is methylated, and then the DNA that has just been so replication errors, correction of replication errors. Yes. I wasn't even thinking of that, but yes. We do that at the end of the semester. Okay, in bacteria though, that is also a way to know DNA repair. Okay. So it's actually direct methylation of DNA. So this is adding a methyl group. Who took orgo? Okay. Adding a methyl group to your cytosine to get 5-methylcytosine. Your cytosines are methylated. For this function, okay? And yes, so it is another mechanism for regulation of gene expression. And so it's done by DNA methyltransferase, another enzyme we mentioned before, and it adds the methyl group to the cytosine. So your cytosines are methylated. And it actually has to flip the base out. And so you can see here in red, your base is actually being flipped out of your double helix, and it's modified. And it's only added to cytosine for this function. But I want to with DNA for this function, yes, only cytosine. And so in order to keep the structure of your DNA stable, which is actually fairly stable anyways, the enzyme sticks a side chain from its own structure as a placeholder while the methylation is taking place. Well, I must be talking. I'm inducing people to take a break. Seeing a lot of people getting up, taking walks. Okay, so DNA patterns, DNA methylation patterns, they're inherited from after DNA replication to the daughter cells through the activity of a maintenance methyltransferase. So this is a DNA methyltransferase that keeps those cytosines methylated after replication, right? Because when you replicate your DNA, you have one new strand of DNA, which is not methylated. And so what this enzyme does is it recognizes, it's a methylating enzyme that recognizes the methyl groups on the parent strand and methylates the new strand of DNA in the same location. And it acts on CG base pairs.
CGUH island?
Hmm?
CGUH island?
CG, I don't know. I think it's just, there are GC-rich islands, but in this case, maintenance methyltransferase is recognizing CG pairs and methylating the cytosine. Yes?
I somewhat remember learning about methylation and deamination. Is there a connection there?
So methylation and deamination. Well, methylation is methylating. Deamination, that's CDU. So that's usually with RNA editing. You'll see that. You must be thinking about it just in the context of alterations to nucleic acid, maybe. Any other questions? I thought I saw a hand going up.
I don't really understand.
Oh, I'm going to walk you through.
Okay.
So here's your DNA before you replicate it, right? And it's got some methylated cytosines, and that's your CH3s coming off of your cytosines. And then you get DNA replication, and these two strands are going to split apart, and they'll maintain their methylation, but then the new strand of DNA won't have it. And so the maintenance methylase is going to recognize the other strand, the new strand of DNA, and methylate it. So it recognizes up here and adds your methyl group.
But is it only where it was already methylated?
Exactly. It recognizes the methylation on the parent strand and adds it because it's CG pairs, and so that's still going to be CG pairs on the daughter strand. So it's going to recognize the methylated cytosine in the CG pair and then add it to the daughter strand. Okay, and so that's how it's maintained through generations. Questions? Okay, so in bacteria, methylation is used to identify newly replicated DNA to repair, so they also can repair DNA after replication, and also to identify phage DNA. Because remember, bacteria get infected by bacteriophage, and they have their own DNA, and their DNA is not methylated, so it's a defense mechanism that bacteria have, and they have these enzymes that can, endonucleases that can recognize and cleave unmethylated DNA in bacteria. Questions?
Are you called restriction enzymes?
Yeah, so that's how some restriction enzymes came to be. Yep, there are specific, there are a lot of restriction enzymes that were identified this way. Yep. So you can see one is this HHA1 is the one that methylates, and then there's also an HHA1 endonuclease, which
So phage DNA is not part of bacteria?
phage DNA is bacteriophage. So who here has learned about bacteriophage before? Or is it's a little bit foreign to some people. So bacteriophage are just viruses that infect bacteria. And so similar to a virus that infects us, viruses bring in their own genome. That's not our genome. And we also have mechanisms to recognize viral DNA and destroy it. And so bacteria have mechanisms to recognize phage DNA and destroy it. And one of those mechanisms is recognizing unmethylated double-stranded DNA. And they have endonucleases that recognize and degrade it. Okay. So in plants and mammals, DNA methylation is associated with gene silencing. So can you propose a mechanism? How is how does DNA methylation, so you've probably learned a million times DNA methylation is associated with silencing. How? What is it doing? Yeah.
It's just putting those methyl group and so it's just completely like blocking the pathway for like DNA polymerase. It can't really sit down because it's just like making these bumps. So
Okay.
DNA is not exposed to.
So it's preventing interaction with RNA polymerase. Yeah.
And also, there's a polymerase that's already attached to the strand and it's moving down the strand and the methylation process.
So if there's a polymerase that's already attached and it's transcribing, the methylation will cause the polymerase to fall off. What else? These are all right. What else is how else is methylation? Yeah.
Wouldn't also be all DNA being methylate and it starts to condense, and then because of the RNA will be what are
Okay, so prevent it. So since it's associated with condensed DNA already, RNA polymerase can attach to the promoter. What else?
Maybe it's a charge. If it's a positive charge. Another one, like RNA polymerase also has a positive charge.
Okay, is the charge affecting things? You know, this came up last year.
you say like preventing DNA.
Huh?
Would it be the same like preventing DNA? It's covered in charge.
Yeah. So let's stay away from the charge for a minute. But let's talk about other things that might be recruited to methylation, right? So you'll have, just like methylated histones are going to recruit other proteins, you'll also get recruitment of proteins specifically to condensed methylated DNA that will prevent transcription factors and RNA polymerase from binding. Well, chromodomains recognize methylated lysine, whereas here it's methylated DNA. Yeah. So think about all the types of proteins that might bind to methylated cytosine and what kinds of proteins that we've learned about that may have a silencing effect. But we've kind of talked about those things. I believe I have, yeah, I've got a few figures to kind of show this to you guys. Right, so here you can have this right here showing the methylation on the DNA that's wrapped around the histone, and you might recruit some kind of protein that contains a DNA methyl binding domain, which is different from a chromodomain. It binds to methylated DNA. And that might recruit a histone deacetylase, an HDAC. Is that logical? That HDAC will then remove acetyl groups from neighboring histones, right? That's all associated with silencing, right? So always ask yourself, is this promoting silencing? Is this promoting activation? You might also get another protein that has a DNA methyl binding domain but is a histomethyltransferase. And so that'll get recruited to methylated DNA and then will methylate surrounding histones, which are involved in condensation of DNA and creating repressive chromatin. You might also get, well, so this is another thing that might happen. So you might have a chromodomain binding protein that then might recruit a DNA methyl transfer. So this is just another form of silencing that induces DNA methylation. So you might have a chromodomain protein that binds to methylated histone tails and then recruits a DNA methyl transferase and then methylates the DNA in that region. That's associated with silencing. And then you also might have a protein that recognizes methylated DNA that's going to recruit a nucleosome remodeling enzyme. Again, associated with silencing. So you can actually think about the practice problem we just had with the viral protein that had the bromodomain, but you can kind of change it change it in your mind to chromodomain, and it might be a protein that's going to recruit a nucleosome remodeling enzyme that's associated with silencing. And so this is kind of big picture. This is from right out of the textbook. I like this. So you can have these histone reader and writer proteins that are all working together to establish repressive chromatin, and they're doing multiple things, right? So it's like what we've been talking about, where you'll have some gene regulatory protein recruit some writer protein that's involved in methylating, let's say, histones, and then recruiting these reader proteins that see those methylated histones and recruit more writer proteins that methylate histones, and then that might recruit a DNA methylase, which then will start methylating the DNA. And then all those methylated DNA will be recognized by some DNA methyl binding protein, which will further inhibit recognition and access to the DNA, creating repressive chromatin. So you can think of these as all working together. Any questions about this? Okay, so I'm going to end with this slide. I just want you to kind of think about bringing all this together. I've given you a lot of little bits and pieces, and I've been trying to bring it together. I want you to be able to connect your histone modifications with your recruiting proteins, which either have your activating or your repressing activity, and you can decide as you think about it which direction to go in, right? If your histone modification is acetylation, it's going to be more activating, recruit those kind of proteins. And if it's methylation, it'll be more repressing and recruit those kinds of proteins, right? And then connect that with DNA methylation and how it all affects gene expression in response to an outside signal. Okay, that's your big picture that I leave you with to think about. If you would like, we have another 15 minutes. You can discuss with your neighbors. And I'm here to answer any questions, or you can turn away. Yeah. Otherwise, it'll be like